Why does the same antibody therapy work wonders in one patient, but not in another? If you've ever wondered why immune responses can be so unpredictable, it's time to dig into the world of Fc receptors and the genetic quirks that make each of us unique. Fc gamma receptors, those little proteins on immune cells that latch onto the tail end of antibodies, are a lot more complicated than you might think. The genes that code for these receptors, called FcGR genes, are packed with variation. We're talking single nucleotide polymorphisms, tiny DNA changes, and even differences in gene copy number. These aren't just minor tweaks, they can completely change how much of a receptor gets made, how well it grabs onto an antibody, and what sort of signal it sends to the rest of the immune system. So, what does this mean for immune responses? Let's take antibody-dependent cellular cytotoxicity, or ADCC. If a patient has a version of the FcGR3 receptor with a certain polymorphism, let's say the V158 allele, immune cells might bind therapeutic antibodies with extra strength, amping up the response. Someone else with the F158 version might see a much weaker effect from the same drug. But it's not just one receptor, there are activating and inhibitory Fc gamma receptors, and the balance between them can tip the scales toward inflammation, or toward quieting things down. These genetic differences ripple through the immune system, influencing everything from cancer immunotherapy effectiveness to how well we clear infections, to our risk for autoimmune disease. Researchers are now looking at Fc receptor genotyping as a way to stratify patients, maybe even predict who will benefit most from a specific antibody therapy. Pair that with FcGR expression profiling, and you've got the makings of more personalized medicine, rooted in immunology. Of course, Fc receptor polymorphisms aren't a magic bullet. Clinical outcomes depend on antibody design, the disease you're fighting, the flavors of immune cells in the mix, and the push and pull between activating and inhibitory signals. But here's what's exciting. Understanding Fc receptor genetics is helping us connect the dots, from tiny DNA differences to the big world of immune diversity we see in patients every day. The more we learn, the closer we get to truly tailored immunotherapies. Thanks for watching.